start this off. I did my A-levels at private school. I was there on an academic scholarship, and my A-levels were chemistry, physics, maths, and further maths. I've always much preferred the sciences. That was a very easy decision for me to make, but it's not an easy decision for other people. And then I applied to Trinity College, Oxford, and I'm now studying chemistry. And I'm now doing a master's project. So that's fourth year of my chemistry degree, and I'm doing a master's project. So my whole year for the Oxford chemistry course, my whole year is research. So before I tell you about what I'm doing, we're going to cover mass spectrometry a little bit more vaguely. You may or may not have covered this in your A-level courses. If you're doing A-levels, it's covered in some and not in others. So I'm going to go over it as briefly as possible. Very simply, break it down as much as I can. Mass spectrometry is just a technique to determine what the mass of something is. So you put a sample in, that sample can be a solid, a liquid, or a gas, depending on whatever your machine is. You then generally do turn it into a gas somehow, and you ionize it. Again, this can be done in many ways. You can put a positive charge on it, you can put a negative charge on it, you can put a little proton on it, so it's overall positive and it has an extra hydrogen. But ultimately, a molecule, you ionize it. Some ions aren't very stable. And so sometimes your molecule then breaks up. So for example, if you were to ionize CO2, you've got carbon and 2 oxygens. Depending on your method, you might also make that break up into a single carbon, a single oxygen, and 2 single oxygens. And so the different ionization techniques, you can either choose to use one that makes stuff fragment a lot, and that's kind of called hard ionization, or you can choose a method that tends to keep things in their original form, just with a charge on it, and that's kind of soft ionization. So you have your ions, you then send them through some analyzer, and all this does, there are many ways of doing this, but this just separates things according to their mass, and more specifically, according to their mass to charge ratio. So that is very important later, but things, very big molecules like proteins, it's pretty easy to get, 6, 7, 8, even more charges on them. So it's very important that you try to remember that it analyzes the mass to charge ratio. Most things only have one charge. If you only have a little molecule, only got one charge, so it's normally not an issue. Through an analyzer, and it hits a detector, and you get some sort of readout that you can then tell the mass to charge value. So that's mass spectrometry. What I'm interested in, and what is most often used on an A-level syllabus, is time of flight mass spectrometry. This is where you have your molecules, and you send them through, you ionize them, and then you have an electrode, so just a plate with a voltage on it. And that accelerates your charged particles. So for example, if you have a positive ion and you have a negative electrode, all the positive ions are going to be really strongly attracted to the negative electrode, and it just has a little hole in the center, so they're all shot through the hole. Very simple technique. And the key thing is that when you have a positive ion and an electrode with a fixed voltage, all of the ions with the same charge get the same kinetic energy. So that's this equation I've given you. Ke is kinetic energy, and little z is the charge, so +1, +2, etc. And big V is the voltage on the plate. So all molecules with the same charge get the same kinetic energy. And if you can remember from your physics, kinetic energy is related to mass and velocity by half mv squared. So you can see that if all the ions of the same charge have the same kinetic energy, that means different masses will have different speeds. And that's why this is time of flight mass spectrometry. You make your ions, they fly down a tube, and they will fly at different speeds according to their mass. And then all you need to do is when they hit the detector, you just note down, well, the detector notes down, obviously, what time they hit the detector. And we do the maths backwards, rearrange some equations to figure out what their mass was. So that's time of flight mass spectrometry. And I have, Yes, I do have a little Slido poll for you. So just to keep you fresh, so if you could go to the Slido, I have a little quiz, which is that you have two particles, both of the same charge. One is mass 19 and one is mass 12. And which one hits the detector first? So I'll just give you sort of 30 seconds, a minute, have a think through, and we'll see what you think. No pressure. This is just to see if you know what you're thinking. No pressure at all. You can just make a guess. Right. And we've got
just to confirm, Megan, we've got lots of votes coming through, which is great.
Lovely. I'll give you all just a couple more seconds. So which one hits the detector first? Which one is traveling quickest? Oh, lovely. Thank you. Great guesses coming in. Bit of a split. Interesting to see. Okay. It's not a trick question, I promise. Okay. So if we
will just get that presentation back, Megan.
Yeah, no problem. So was it 70% of people said, 71% of people say mass 12, and if I can do my maths in my head, 29, yep, oh, someone changed it. So 74% of people say mass 12 and 26% of people say mass 19. Just a bit of rearrangement, so not always so easy to do in your head.
It's coming, I promise.
That's okay. It's quite a big file, so
I might relay the next one to you verbally just so we don't have this slight delay.
Yeah, that's okay.
So yeah, 74% mass 12, 26% mass 19. 34 votes in as well. 100% of you voted. Okay. Fantastic.
Wow.
It is coming, I promise. I can see the green line inexorably coming up.
Sorry, everyone. I have lots of pictures, so it's a huge file. There we go. Okay, so if I just...
I will whiz along. Oh, yeah, go on, Megan, you can take it forward.
Can I? Let me just find it. There we go. Okay, lovely. So, mass 12, mass 19. Most of you said mass 12. And so I've just rearranged the equation here. So you can see that larger mass is on the bottom, little v is velocity. So larger mass means slower velocity. So larger mass takes longer to arrive. So those of you who said mass 12, very well done. The lighter masses hit the detector first. I won't make you do any more maths today, don't worry, I promise. But thank you for giving it a go. So just a brief explanation of why on earth we would find this at all interesting is, here's a brief example. So these two molecules have the same chemical formula, C6H12O, but as you can see, they're very, very different structures. These structures, if you've come across them before, they're called skeletal formula. And all it is because when you get to very complex organic molecules, drawing out all of the carbons and hydrogens just gets very complicated and takes a very long time. So every corner is a carbon, and you just add on as many hydrogens as is needed to mean that carbon has 4 bonds. Because generally you just assume that carbon will have 4 bonds. So you can see that they're very different structurally. One's an aldehyde, that's hexanal on the left, and one is an alcohol, cyclohexanol on the right. And this is their mass spectra. So you have the same starting mass, but because they have different structures, when you ionize them, they fragment and they form different ions. So different bits break off, basically, and all the different combinations add up to make this mass spectra, and they're very, very, very different. So if you had a reaction and you knew in order for your equations to balance. You needed to make C6H12O, but you didn't know which one you'd made. You put it in a mass spectrometer and compare your sample to a known sample. So you would test beforehand cyclohexanol and hexanal and say, which one does it look like? So it's a really, really powerful tool for identifying unknowns. And on to the next Slido poll, just to get you thinking again, why on earth do you think this is useful? Why do we even care? You have some slight spoilers, obviously, if you've looked at the description for this talk. But I just want to see what you're thinking, whether you're thinking of the same things I have, whether you've thought of applications I haven't mentioned in this talk. So just to see anything you like, any applications you can think of. So the same Slido poll, the same Slido as before. Lovely. Okay, so we've got some answers coming in.
Would you like me to display, Megan, or would you like to shall I read them out? Or
Yeah, if you want to read them out, that would be great. Thank you.
Probably be a bit more straightforward. I didn't do very well getting that slide back, did I, before?
Just a good file.
So identify isotopes, atmospheric chemistry, astrochemistry, medicine, forensics, identifying products, atmospheric contaminants, identifying pollutants, drug identification, industry, isotopes, lightweight sport equipment, drug testing and sport, test products. Oh, big range of ideas here.
Yeah.
Protein identification, carbon dating. Fantastic. What a range.
Lovely range. Yeah, brilliant.
I think I've got the full range there. Industrial testing. I think I might mention that. Yeah, great.
Lovely. Thank you. Yes. So many things you've mentioned will come up. Some haven't. So I suppose well done if you put something in that I haven't had time to show. There are so many applications. So forensics, absolutely. Someone mentioned forensics. Food and drink industry. Drugs, of course, there's lots of stuff about pharmaceuticals and medicine. Medical diagnosis as well. These are just a few that I've picked out. Obviously, atmospheric and astrochem, as you said, and the environment. So I've just gone into a little bit more detail about a few of those, starting with forensics. Forensics, the key thing with forensics is often identification of compounds. So as many of you said, you know, if you have something on the scene of a crime, if you worry there's a drug there, if there's a poison there, if you don't know what something is, you can test that with mass spectrometry. A lot of these applications are combined with other identification techniques. So it's often not just mass spectrometry, but it's sort of one piece of the puzzle that helps you understand what's going on. So forensics, absolutely vital for forensics. If you want to test if someone's been poisoned, you can check if it's in their blood with mass spectrometry, potentially. Archaeology, as some of you mentioned, carbon dating. It's a huge, huge aspect of archaeology, finding out how old something is. And part of that is, as came up quite a lot, identifying the different isotopes. So you can check how old something is by saying how much, generally carbon-14 is radioactive, how much carbon-14 is in this compared to other isotopes of carbon. And because isotopes are different masses, you can do it with mass spectrometry. And the other thing that I really loved, I actually found this in a book about the Vikings. You can find chemistry everywhere. Book about the Vikings, you can tell from very old bones and skeletons where someone was born and where they grew up, based on the different ratios of isotopes of strontium in their bones. Strontium is a chemical element. It's in pretty much everything. It's in your food. It's in, you know, the rocks and the soil. It's in everything in very, very tiny amounts. And depending on where you are geographically, there are different amounts of each isotope. And when you eat, you know, when you eat the fish, when you eat the plants that have been grown locally, that strontium ends up in your bones and your teeth. So you can check skeletons and say, well, that skeleton, while it was found here, we can tell from the teeth that they were born from somewhere else. Doesn't work quite so well nowadays. Obviously, we eat a lot of food that's imported from other places around the world, but it works very well for archaeology, where people tended to be more stationary and food was grown locally. So I thought that's incredible. You can look at a Viking skeleton, for instance, and say, well, they're buried here, but they didn't grow up here and they weren't born here, so they must have traveled here. It's a way of distinguishing, say, for example, with DNA records. You can tell from the DNA records where someone is from, sort of ethnically, culturally, but you can't tell where they lived. The other thing is the environment. Obviously, there are lots of contaminants in the air, pollution. You know, there might be heavy metal contamination if there's a factory or a mine nearby a river. Some ones I've mentioned in my notes actually are nitrophenols, which are produced from pesticides, and these can make their way into drinking water. So you have to have a way of testing whether or not they're in the drinking water and whether they're at safe levels. Nitrosamines, which I will mention again later, also can often end up in the water supply. And again, you need a way of testing whether or not they're there and how much of them there are. Also, quite an interesting thing is there's sometimes a lot of synthetic estrogen in the water supply. That can be from contraceptives. It can be from, you know, other hormonal treatments such as HRT. If you're undergoing the menopause and you need hormonal replacements, things like this. And in some places, the levels have got high enough that they're worried it's going to start affecting fish fertility. So there are also other ways that you really need to be able to test the levels of these compounds in the water and in the environment. Big one, the environment. Diagnosis. So this is another, you know, some of you already mentioned medicine, and this is a huge thing in medicine. You can actually tell if someone has had a concussion from a blood test. A couple of days after someone has had a concussion, in their blood, this particular sort of biomarker is what we'd call it, pops up. And a biomarker is just a biological molecule that, you know, you can identify as being related to something, essentially. So if your body produces this particular compound in excess if you've had a concussion, so you can find it in blood test. Obviously, it's a bit late at that point, but it's still interesting. Some cancers nowadays can be diagnosed with a blood test. So prostate cancer can be diagnosed by a blood test. Again, just seeing what molecules in your blood and certain levels of certain molecules that you would detect with mass spectrometry indicate that you have prostate cancer. And also it can be used to track how well your treatment is doing. Prenatal screening for certain, you know, for certain infections and things, that can be done with mass spectrometry. You can monitor people's cholesterol levels if they've had a blood test. And for example, COVID antibodies. All of this stuff can be done with mass spectrometry. It's often combined with other techniques, but mass spectrometry forms a key part of those techniques. Huge thing, diagnosis. And food, which might come as a surprise, but this particular example that I found interesting is honey. And honey can contain, I don't know how to pronounce this, sulfonamides, which is just a class of antibiotics. And they're used to treat this particular disease in honeybees. But that means they end up in the honey. And obviously in very high amounts, they can be dangerous to humans, and they can lead to antibiotic resistance. So you need to check whether or not your honey has low enough levels of these compounds. As a similar example, perhaps, allergen testing. Does something contain nuts? Well, how do you find out if something contains nuts? One of the ways you can do that is with mass spectrometry. And finally, say perhaps you were in the shop and you bought a packet of mints and you wanted to check whether or not that contained beef or horse. You could do that with mass spectrometry, and it has been done. And that is, again, just you would take a sample of beef first, take a sample of horse, and then you'd test your mints and say, well, does it look like the one on the left, or does the mass spectrum look like the one on the right? Or is it a mixture of the two? And so from that, you can identify what molecules are present in this meat, and therefore what is it likely to be made of. And finally, research. If you have made something in the lab and you want to check what it is, one of the techniques you can use, very, very often combined with lots of other techniques, is mass spectrometry. And perhaps more specifically, I know someone mentioned, protein identification. Mass spectrometry is a very important technique for protein identification as well. Have you made the right thing? Have you got enough of it? Has it folded in the right arrangement? All of these things affect the mass spectrum, and therefore you can check whether you've made the right thing. You can also study the kinetics, so the rate of certain processes of drug metabolism, how your body processes the drug, how quickly. You can do this with a blood test, or you can measure the amounts of certain molecules in your breath. And one of the ways you can do that is with mass spectrometry. So it's a really, really powerful technique. I realise I've covered most of modern society, but that's because it is found in most of modern society. And the final bit of my talk is going to be more specifically about what I'm doing and what the Valence group is doing as a whole. So here's the Valence group. Here we are pictured. Claire Valence is the principal investigator. So she's sort of the head of the group, and she's in the middle at the front row. If you're very eagle-eyed, you might be able to spot me at the back, peeking between two heads. And as part of the chemistry undergraduate course at Oxford, as I said earlier, your entire fourth year is a research project. So I'm just working in the group full-time, sort of a full-time job really, working alongside the PhD students and the postdoctoral students in the group. And one of the things the Valence group is looking at this year are pharmaceuticals. And I mentioned earlier nitrosamines. On the screen pictured are just a few nitrosamines. It's just a class of compound. The key thing being they all have this nitrogen-nitrogen bond and an oxygen. And these are a very common contaminant in certain classes of drugs. But in too high a level, they can be carcinogenic. So these drugs have to be tested very, very carefully and very regularly for the levels of nitrosamines they contain. And one of the ways that that can be tested is with mass spectrometry. The second thing the Valence group are looking at are clinical applications of mass spectrometry. So this is, I think this is incredible. You can use machine learning algorithms. So that's where you sort of train a computer to build a model to fit your data and then predict a result based on the previous data that you've given it. You can use machine learning to identify sort of mass spectra. So different tissues in your body, for example, in this human brain sample, the brain contains lots of different tissues, and they will all have a unique mass spectrum. And they'll probably look very, very, very similar to the human eye. And that's why we use machine learning to do it. And if you can feed a bunch of mass spectra into your code, and you tell it this mass spectrum equals this tissue, and you do that for enough pieces of data, eventually you'll then be able to get a model where you can give it an unknown mass spectrum and it will tell you that's this tissue. So on the left is just a normal imaged slide. This is how you would normally look at brain tissue. And on the right is the predicted grouped mass spectra with a machine learning algorithm. So you can see it does a pretty good job of matching the two up. And the idea would be is that, say, real time in surgery, you would be able to have some sort of little knife or a pen or something or a probe. You'd be able to touch to the brain tissue, and it would tell you that's healthy tissue, leave it alone. Or it would tell you that's, for example, tumor tissue, cut it out. Because obviously you don't want to be leaving tumor tissue in the brain if you can, because then it might lead to it growing again and coming back worse often. But you also don't want to cut away healthy brain tissue. So that would be the eventual idea, is something that would be able to identify real time, is that healthy tissue or is that tumour tissue? And it's identified with mass spectrometry. And finally, don't worry, I haven't forgotten about it. Finally, astrochemistry. This is another huge application, in terms of atmospheric chemistry, what pollutants are in the air, in terms of astrochemistry. For example, the Mars exploration, they would send up the Mars rover with a mass spectrometer on board, so that any air, any gas, any soil, any rocks, any liquids they find on the surface can be identified. And as another example, if you saw the news article about potentially finding life on Venus, phosphines were identified as a molecule of biological interest. And again, combined with other techniques, but one of the ways that you can identify are there phosphines present is with mass spectrometry. Hugely powerful tool. And what I specifically do is time of flight mass spectrometry. This is what our equipment actually looks like. We do not use a commercial mass spectrometer because of the kind of things that we're doing. So this has been built by other members of the group. But to make it simple, you have molecules traveling along the middle. We ionize them with electrons and they carry forward to hit the detector. Ignore that's all that metal tubing is doing. The reason why there's all those wires and tubes is just because we do it in a very high vacuum. So that's all that's there for. Essentially, it's exactly the same as the previous picture. And what I'm looking at is actually radiation damage. So, for example, when you are exposed to the sun, you get a sunburn. The molecules in the top layer of your skin are damaged. They absorb the UV radiation, for example, but then they release electrons. And it's actually those electrons that go on to cause more damage in your body. And the overall idea would be if we can look at how, for example, proteins or your DNA interacts with those electrons, we can understand radiation damage a lot better. We could maybe better track it to know how bad the exposure was, to better treat it and to better manage it. That is a protein pictured there. I am not working on proteins with the kind of data that we get. If I were to put a protein in that machine, it would just fall into millions of pieces, and I would just get sort of this sea of stuff hitting the detector, and I would get no usable information from it because it would be way too complicated. So I'm studying pretty much the simplest analog you can, which is DMF, N,N-dimethylformamide. That is nitrogen, and it's got two CH3 groups, which are just those sticks coming off it. And then it has a COH group, which is that section coming off the left. And this is a mass spectrum of DMF. So the idea would be is if we can look at how DMF acts, it has a peptide bond, which is that nitrogen to carbon to CO bond. That's peptide bond. If we can look at how this acts when we hit it with electrons, we can eventually extrapolate that quite a bit further to maybe try and guess how a big protein containing millions of these peptide bonds would act. And DMF and other molecules that the group study are found in the atmosphere and in sort of the interstellar medium in space. And so by studying these molecules, seeing what happens when they're exposed to electrons, just like they are in the atmosphere and in space, we can form better models to model the atmosphere and to model space and to understand what's going on. So that's what I do. And final slide, this is the end. Final slide is just some acknowledgements. The whole of the Valence group, obviously, I'd really, really recommend if you enjoy any of these applications to go look at the group website, which you can find on chem.ox.ac.uk if you go to Claire Valence, or if you want to go straight to the website, it's valence.chem.ox.ac.uk. So the whole valence group, this is just pictured as a pottery that we painted over Christmas as a social activity, and my parents' cat for being really cute. So that's it from me. And if you have any questions, please do let me know. And otherwise, I can just talk more about chemistry and why I chose it and how I got here.
Great. Okay, Megan, first question come in. What is the most exciting prospect, in your opinion, of mass spectrometry?
I think the clinical applications of mass spectrometry are really only just being realised. There are so many things that you can detect from a blood test. You know, as I said earlier, concussion. I never would have thought you can detect a concussion with a blood test, but so many diseases you can monitor. You know, the outcomes of patients if they've had a heart attack with a blood test. There's so much potential in terms of disease monitoring, because a lot of the problem with treating disease is, for example, with heart attacks, not everyone needs surgery. And you don't want to make people have surgery if it's not necessary, because there are additional risks associated. So combining mass spectrometry with this machine learning, I think is going to be really, really powerful in the future for really efficient and effective treatment of diseases.
Could I ask one, please, Megan, relating to something that's been a frequently asked question through this series in different disciplines, obviously. If you can think back to the time when you were, say, in year 12, any particular supercurricular resources you'd like to recommend, something that came on your radar then that sort of inspired you or made you think a bit?
I think there's this particular book that I did really enjoy called Napoleon's Buttons. I don't remember the author, but it should come up if you Google it. It's just a really nice sort of series of interesting chemistry uses and applications. But I mean, otherwise, generally, I mentioned YouTube channels on my personal statement, things like SciShow, I'm sure you've all heard of. So don't feel like what you mention has to be super academic or fancy. I talked about YouTube channels. I talked about this BBC documentary I found on poisons. So you know, you really don't have to mention anything super academic.
And apologies to the person who asked that question. It was a bit related to mine because I pipped you there, but yours is more specific. What uni course and extra or supercurricular activities would you suggest for a career in astrochemistry regarding mass spectrometry? So I suppose the same question, but related more specifically. Did you read anything supercurricular wise when you were pre-university about mass spectrometry? I'm still having problems saying that, but you know what I'm trying to say.
It's a bit of a tongue teaser, honestly. I've had to say it a lot, so I'm used to it now. I don't think I read anything sort of mass spectrometry specific. That one was even worse. I think, you know, stuff like carbon dating definitely came up. But I don't think I read anything that was specific to astrochemistry or mass spectrometry. It's just something that I've discovered throughout my chemistry course and just in doing this master's project that I really enjoy.
And so, okay, I'll twist the question slightly. Not before you applied to uni then, anything that you've read in the course of your current studies relating to astrochemistry or mass spectrometry that is sort of relatively accessible that you'd recommend? I mean, I guess you read loads of things that are very technical in the course of your research, but anything that's sort of perhaps more accessible, wider pitch?
I mean, I think so there are definitely documentaries on, you know, space. I'm sure the Brian Cox ones you will have all heard of. There is, I can't remember the name of it, but I think there was a BBC documentary about the sort of discovery of phosphines on Venus that might be of interest to you. And I think just little things like that. There was definitely, I mean, the book I mentioned on the Vikings, that mentioned carbon dating and this strontium the strontium sort of isotope mass spectrometry, and that wasn't a specific chemistry book. That was a history book, and I found the chemistry in it. So you can sort of watch just more general Brian Cox documentaries, or a documentary about how they discovered all these phosphines, but you can find chemistry in pretty much everything.
Another question, what techniques do you use alongside mass spectrometry?
So alongside mass spectrometry, there's a lot of spectroscopy, which I always get muddled up, but spectroscopy means that it involves light of some form. So UV radiation or infrared radiation or microwaves. So it's a lot of spectroscopy. Things like IR spectroscopy, if you've covered that in your courses, where you look at the vibrations of bonds in your molecules. They're a really good way of identifying molecules. You might not have come across it, but Raman spectroscopy, very similar to IR, but rather than looking at vibrations, you're looking at rotations, how the molecule rotates. Things like UV-Vis spectroscopy, where you look at how much of visible light or UV light something absorbs. That's a really great identification technique. NMR, if you've covered that in your chemistry courses yet, that's a really, really powerful tool. So lots and lots of other techniques are often combined, particularly in research labs, to identify these compounds.
Is mass spectrometry one of the best ways to diagnose people with illnesses? For example, is it the most effective? I suppose is it effective? I suppose as well.
In terms of, I'd say mass spectrometry isn't alone the most effective way, and it's certainly not the most effective way yet. I think we've got quite a few years until we can, have a test for everything that you just pop it in a mass spectrometer and it says, yep, they've got high cholesterol. These are the tablets you give them. I think it's a useful tool, but it's nowhere near the best thing we have at the moment. But I do think it has the opportunity to be combined with these incredible machine learning methods. But currently, I think, the normal diagnosis techniques that doctors use every day are much more effective.
And then a bit moving on towards your own experiences and trajectory, as another word that I always find difficult to pronounce. What course did you apply to uni with? So at the start, Megan, you mentioned that you applied to university doing 4 A-levels, and they were further maths, physics and chemistry. And then what course did you actually apply for at Oxford? Was it chemistry or?
Yeah, so I applied to chemistry at Oxford, and the Oxford chemistry course, you have to do 4 years, so it's a master's course. And as I said, the whole final year is the master's course, but there are lots of other chemistry courses at other universities where you can choose to do 3 years and then do a master's at another university, or not do a master's at all. But I applied to do sort of pure chemistry.
Without going into too much detail, earlier on I mentioned I was at a college in Durham today, speaking with year 12s, and generalising, I was saying generally university courses across a wide range of disciplines, you've got a kind of fairly fixed or not that much flexibility in first year, for instance, for what you take. There's various things universities want to teach you that are common to all. And then there's quite a lot more optionality as you go forward. And obviously the Oxford degree is a 4-year one. Do you get quite a lot more choice as time goes on, for instance, in the Oxford degree?
So yeah, it's definitely the first year for the Oxford degree is completely fixed. Some universities you have sort of 1 or 2 options, but the Oxford course is completely fixed. And the second year is almost completely fixed. You could choose to do what we call a supplementary subject, which is just you just do an extra exam, and that gets you a little bit of extra credit. So you don't have to do so many labs if, like me, you don't like lab work so much. And then in your third year, you do get a lot more choice. And so you have your first term is all fixed courses, and your entire second term is optional modules. So you just choose whatever you want to study out of a list of, I don't know, 25 or so of them. And then what I loved about this course is that obviously your fourth year is completely up to you. The chemistry department publish a list of all of the academics in the department who have space to take a fourth year student. And I mean, it's normally most of them. So you can have a look on the department website if you want to see how many options that is, but it's a lot of options. And you can even apply outside of the chemistry department if you want. So some people apply to work sort of in a biochemistry lab or a biology lab, and it still needs to be a chemistry related project, but it doesn't necessarily have to be with a supervisor in the chemistry department, as long as the department sort of makes sure that there's enough chemistry in your project. So you have an enormous amount of choice, and you sort of just, you rank sort of 1, 2, 3, these are my choices, and everyone gets allocated their choices. So there's definitely a lot more choice as you go through the course. The Oxford course generally, I'd say, has less choice than other chemistry courses. And that is actually why I picked it, because I had no idea what I wanted to do, so I wanted to do everything. But that is something to be aware of. It does have less choice than other courses. So if you want to do something super specialised really early on, just make sure that is available to you, whatever course you choose.
And another one come up. I was quite surprised to hear you say that you're not so keen on lab work because you've been so infectiously enthusiastic about it. How much time do you typically spend in labs in the Oxford degree?
So in your first 3 years, for any chemistry degree that's accredited by the Royal Society of Chemistry, you have to spend 400 hours in labs. And so every university that has a chemistry course that's accredited, which hopefully they will be, is 400 hours minimum. And the Oxford course has 400 hours minimum. So it's a lot of lab time. Some universities have more than 400 hours. And so in your first year, you do 180 of those lab hours, and then the rest is in your second or third year. And that's for the Oxford course. You spend 2 days a week, and then you get every so often you get a week off. So in your first year, I think it's 3 weeks with labs and 1 week without. And in your second year, it's slightly fewer labs, and in your third year, slightly fewer.
So the 180 hours in the first year of the course is 24 weeks, 3 Oxford terms, and I guess that's something like 7.5 hours a week, which you kind of split over a couple of, right? Yeah.
Yeah. So it's 2 days a week, 11 till 5, you're in labs.
And then it's actually proportionately less in the latter 3 years then. Yeah, that's kind of surprising, isn't it?
Just in your third year, it means that you can hopefully get your lab hours over before you, so it gives you more time to revise for your third year exams that are obviously more important. But so it feels like a lot of labs, and I don't like labs so much, which is why I'm doing the master's project that I am, because as much as I'm in the lab, it doesn't really feel like a lab. It feels more like a workshop. We've got spanners and workbenches and all this sort of, machine parts and electrical equipment. So I don't like being in labs that much. So that's why I love this, because it doesn't feel like I'm in a lab. But if you want to be in a lab, you absolutely can be.
We've got another one. What were your Oxford interviews like?
Yeah, great question. Very scary. I don't want to lie. I did find them really scary, and I did find them quite difficult. I think I'm sure Richard's heard this a lot of times now, because I tell everyone, as hopefully to reassure you, in that I thought they went very, very badly, because in one of my interviews I forgot how many bonds nitrogen formed, which is quite important for a chemistry degree to know how many bonds nitrogen and carbon normally form. And I just completely blanked. and I had no idea. And I was sort of like, oh, I took the 4. I don't know. But I walked out of the interview going, that is the worst thing I've ever done. That was so embarrassing. They're going to think I'm really stupid. And I also got, halfway through a question, I saw the question, I went, oh, I just did this the other week in school, actually. I really love this topic. I got halfway through and realised it had not been something I'd done. It just looked kind of similar, but I'd really hyped myself up. And now I had to say, actually, I've never seen this before in my life. I've no idea what I'm doing. Can I have some help, please? So, I felt like I'd done so badly, but it turned out okay.
Yeah, I'm just thinking, you said sort of scary and potentially sort of daunting, but I'm guessing that actually, and you're very modestly telling us that you had a couple of things that might have seemed to have gone better, putting it mildly. But I'm guessing that there's somebody there to sort of, metaphorically speaking, hold your hand through the process and say, okay, let's look at this from another, if you think it went really badly, did somebody kind of help you turn it around in the interview?
Yeah, absolutely. I mean, the tutors, they have done this a lot, people who are interviewing you. And so they're very good at they will give you hints. So when I said, actually, I haven't seen this before, I said, okay, here's how to start thinking about it. And they will give you hints because they want to see you, they want to see how you think, they want to see you get to the right answer if you can. And so they're not going to leave you stranded. They did jump in and say, okay, what about this? And so it can feel like it went really badly, but ultimately they just want to see that you can take hints and you're a good person to teach. So even if you don't remember much chemistry, you can show them that you can, take the feedback and improve.
So thank you to our guests for all their cracking questions. I'm going to add to that previous question with another one about interviews. In chemistry, how much would you say it's about knowledge in the interview and how much is about thinking skills? So, for example, was the nitrogen related question a kind of warm up, or was it a typical question of effectively working with your knowledge?
So the nitrogen question, for example, that was, I think, kind of partway through the interviews. That was sort of, we were in the flow, and it was just, that wasn't the question, how many bonds does nitrogen form, but it was sort of a reaction scheme in which you needed to know how many bonds nitrogen formed. There is some knowledge that you're expected to know. You'd probably be expected to be comfortable that carbon normally forms 4 bonds, and so you don't accidentally add a 5th on. That's really bad news. But you'd be expected to be comfortable with some, the core chemistry topics. But obviously, every chemistry course is different. Every school teaches things in a different order. So the tutors can't rely too much on you memorizing facts. And a lot more of the interview is focused on how do you think, how do you deal with being presented with information you've never seen before in your life. There were lots of things in the interview that I'd never seen before, and I said that, and I said I've never seen this before, and they said, okay, give it a go. They just want to see how you cope with new material, because that's what you'll be doing in your degree. You'll be being given material you haven't seen before, you don't actually know how to answer, and you just need to give it a go, and then they can help you.
So what's the best way, would you say, and that sounds very similar to admissions tests, by the way, presenting with new material you maybe haven't seen before. What's the best way to kind of prepare for that, preparing for being presented with unseen material? What would you say?
I think it is very difficult, but what really helps is practicing talking out loud and sort of kind of saying your thoughts out loud. It feels really uncomfortable and it feels very awkward and it feels very weird, kind of rambling on. I'm very good at talking, but it does still feel very weird. Just rambling on about here's what I think about this, and I'm not sure about that, but maybe it could be this, or it could be similar to this thing I've seen before. So maybe just practicing talking through your subject out loud, if that's to a parent or a friend or someone else who you can just explain a topic to, and just practice talking out loud.
That's definitely one of the things we recommend in interview workshops. Yeah, talking through the subject, forming little discussion groups. Brilliant. Megan, thank you so much for taking us through this topic tonight. It's obviously got tons of useful, universal as well, applications. Lots of food for for thought there. Thank you to our guests for all their fantastic questions. I love the way there was a mix of sort of questions about chemistry and also questions about, well, approaching higher education and Megan's own experiences. So thank you from us. Megan, I'll leave you with the concluding words.
Yeah, thank you everyone for coming. I hope that you found this useful or interesting, if nothing else. And as Richard mentioned, if you have more questions, you can always chat to the ambassadors online.
Brilliant, thanks very much. And I will send out the PDF of all of these slides and also a recording of the academic part of the session probably tomorrow morning. Thanks very much for joining us. Cheerio.
Thank you.